All right, so last time, actually, let me just make it into full screen. By the way, any quick question regarding project number 2, besides the one on Classum? I guess not. Okay, that's fine then. All right, so last time we looked at different types of medical images, like CT scans, fundus images, skin photos, MRIs, X-rays, and how they are different from one another. And sometimes they could be videos or sequence of images constructing a 3D image instead of 2D images. And then we looked at the basics, which is image classification. So it is not too different from just plain old image classifications that we used to do, or that we are familiar with, such as ImageNet Challenge, or those kind of, or MNIST or CIFAR. It's just given a 2D image, you want to classify whether there is a disease or not. And sometimes it could be multi-label classification because there could be multiple diseases per image. It could be multi-class classification. There can be only one class can be true, or it could be just binary classification, like there is a disease or not. So, and we looked at 2 representative examples, which are Google's retinopathy. So Google's diabetic retinopathy detection model, which was based on Inception version 3 and trained on like hundreds of thousands of images, hundreds of thousands of fundus images, to be more specific, and performing on par with average, average US licensed eye doctors. And another was skin cancer detection from Stanford, also based on Inception version 3, and also trained on hundreds of thousands of skin images, like skin lesion photographs, and also on par or slightly outperforming United States US licensed average dermatologists, skin doctors. So that was. And the important thing was, or the interesting thing was, there was nothing too much clinical in terms of the approach. So they just took a bunch of photos and labeled it. So the labeling process requires some domain knowledge. Like it's not like labeling or annotating just plain or ordinary images. It's not like you are a labeler and you're labeling whether there's a dog or a cat or bicycle or a car in the image. You're dealing with clinical images, so you have to know, you need to possess certain amount of domain knowledge. Like if you want to label for retinopathy, then you need to be able to actually see a fundus photograph and tell whether whether the patient has retinopathy or some other like macular edema or something like that. Like you need to know the disease in order to label. So that is where all the domain knowledge is injected, but the modeling process was not so much clinical. So even like as for the fundus images, as for the diabetic retinopathy, it's just bunch of images poured into Inception version 3, trained with binary classification, cross entropy, and that was it. So that much we can do. It's the labeling that we cannot do. And as for the skin cancer image, the skin cancer detection, obviously labeling process requires some domain knowledge. Like domain dermatologists were involved in labeling the images. But on top of that, in addition to that, the labels were actually curated or constructed a new skin cancer or skin disease taxonomy, like like a hierarchy, so that they could actually group 2,000 different like really granular images into about 800 images. 2,000 granular diseases, like skin diseases, like just plain old like warts or skin cancer, whatever. So 2,000 very granular skin diseases into about 800 skin diseases, and then they aggregated those 800 nodes into upper layer nodes in order to classify whether the image is benign or malignant. Malignant. So on top of the labeling process, the output classification, the output head aggregation process required some amount of domain knowledge. But that was it. Again, like training the model or the architecture of the model didn't have anything to do with eye disease or skin disease. It was just inception version 3. So that much tells how powerful deep learning models can be. It requires zero domain, like the modeling process or the training process requires zero domain knowledge. So anybody can do that. Like as long as people have them, as long as you have the images and you have the GPU server to train the model, then you're good to go. So all right, so that was image classification. So today, as I mentioned on Thursday, we're going to talk about image segmentation. So this might require some amount of domain knowledge, but again, not too much though. So we'll see. So before we go into image segmentation, there are some couple of differences between different types of image applications, such as like there's image classification, there's object detection, there's semantic segmentation, there's instance segmentation. Like we'll just take a look at them one by one. So image classification, we all know what it is. It's we just have an image and then we want to classify a given image into a class or a label. So there is like a set of discrete labels. It's not a continuous regression. It's there's like dog, cat, truck, plane, like here. And then what we want to do is just given an image, we're going to assume there is one object or one class per image and try to classify the given image into one of those classes. That's it. By the way, so all the material from all these images are borrowed from a very nice blog from this website. So if you're interested in a bit more detail, you can visit the website. So the next one is localization. So classification plus localization. So localization is trying to figure out where the content or the object is located inside a given image. So it's not just you're doing more than just simple classification. So give a classification is given this image, you want to classify this into a cat label. But if you want to localize as well, then you want to somehow tell where that cat is in the image. And you can do this by having a boundary like this. So this would be a bounding box. So in order to actually predict or produce the bounding box, you need a X and Y coordinate of this corner and this corner. So you would need 4 scalar values on top of the class prediction. So X and Y coordinate of this, X and Y coordinate of this. So you need like 4 more scalar heads. And obviously you can't do more than just, this like bounding box is not the only way to localize an object. You can do like I'm not sure if you can call like attention or grad chem type of thing as a localization, but you can still tell why the model thinks the image is a cat, and then try to use the gradient based methods or some kind of like a layer wise propagation type of methods, relevance propagation type of methods to tell where the object is. So again, the assumption is one object per image. So there is only one object inside an image and one bounding box of the target object. So object detection is a bit more generalized. So there are multiple objects or multiple classes per image. And obviously you want to have a bounding box for each of them. So multiple bounding boxes or multiple localizations of the target object. So you are all familiar with this. This is what you can do with like Pascal-Bach or MS Coco, that kind of data set. Right. So the next one is semantic segmentation. This is what we're going to do today. So semantic segmentation is the assumption is the same. So like object detection, multiple objects per image and multiple objects per image. So that much is the same. But the difference is that instead of having a bounding box, we want to classify every single pixel to its corresponding correct classes. So assume that this is about like 256 by 128 resolution image. Then you have about how many? You have about so what's that? It's 2 to the 8 times 2 to the 7. So that's about 30,000 pixels. So yeah. So if this is 256 and this is 128, then you have about 30,000 pixels. So you want to classify every single one of each of 31. Each pixel of the entire 30,000 pixels into the correct class. And there are 3 classes here. So there's person, and then there's bicycle, and then there's a background. So you want all the pixels here to be a background, and all the pixels inside this human body, obviously you want them to be classified as person, and all the pixels, including the handles, the wheels, the pedals, like you want them to be classified into a bicycle class. So that you basically need a 30,000 different times of classification. So that's a lot of processing. So you can imagine the output space is gigantic compared to here. Like, you know, like given an image, usually you would have about 5 to 10 objects, maybe 20 objects maximum. But so in that case, you need actually that depends on how object detection depends on what kind of model you use. If you use like fast RCNN, then that kind of requires a lot of processing. But if you just think of this for particular example, if you assume that there's one object and one bounding box, then all you need is one output class and 4 XY coordinates. So that's it. But if you are doing this, you need 30,000 different outputs for each pixel. So that is a lot of output space. There's a gigantic output space. All right, so further on. So there is something called instance segmentation. Which is, the assumption is still the same. So there could be multiple objects per image. And the difference between semantic segmentation and instance segmentation is that you want to classify each pixel into each instances. So there are 3 persons, and 1 horse in the image, and you want to classify or label each person into its own category. So unlike this here, like in this image, you can't actually distinguish between this person and this person and this person, because the pixels that compose these 3 persons are all categorized into the same class. So for example, here they are a bit, they're a bit connected. So in this case, you can't really tell like the boundary between different people. But here, even if they were connected, you would still have to classify each person into different labels. So this is a bit more challenging compared to semantic segmentation, obviously. And if you think about like medical application of semantic segmentation, for example, if you want to segment or recognize different types of organs in your body, like in your abdomen, then you probably don't need instance segmentation. All you need is semantic segmentation, because you only have one organ in your, like one unique, different types of unique organs in your body. It's not like you have 2 livers, right? Or it's not like you have I don't know, 2 hearts. You have only 1 heart, 1 liver, 1 left lung, 1 right lung, and so on and so forth. So this is probably enough. You don't need this inside your body. All right. Yeah, so this is the main difference between when there are multiple objects, what is the difference between object detection, semantic segmentation, instance segmentation? This clearly captures the difference between the 3 tasks pretty succinctly. So, object detection, the output, the expected output is that a bounding box for different objects, and in semantic segmentation, your expected output is a pixel classified into different classes. In instance segmentation, your expected outcome is pixels classified into different instances. Right. Yeah, before we move on to the actual medical application, so why interested in semantic segmentation? Actually, before we go into this, so any question regarding different types of image applications, image related tasks, like image classification, localization, object detection, semantic, or instance segmentation? Okay, there was a chat. Why is it called semantic segmentation? That actually is a good question. I guess that's because you want the outcome, or you want your image to be segmented based on the meaning, or actually, yeah, that is a very tricky question. Yeah, I would think that because you want your it's an object. I guess the more technical term would be object segmentation, but then you are trying to segment your objects inside an image based on their contents, like, so the contents is like the meaning, it's like content of the image is like the meaning of the image, and then semantic means like meaning. So I guess that's kind of related. Yeah. 재원님, 세만틱이나 인스턴트 세그멘테이션 모델의 아웃풋 사이즈가 픽셀 사이즈가 되네요. 네, 재원님의 질문이 정확히 맞습니다. 인스턴트 세그멘테이션 모델의 아웃풋 사이즈는 인풋 사이즈랑 아웃풋 사이즈가 똑같아야 돼요. 테크니컬리. 그렇지 않을 때도 있는데 사실 그냥 직관적으로 생각하면 똑같아야겠죠. 그래야지 픽셀 하나하나를 다 색칠한 게 되는 거니까. 네. So the question from Jaewon Lee is pretty important. So the question was that in either semantic segmentation or instance segmentation, the output size of the model should be the same as the input size, and that is, the question is correct. So if you want to color, if you can think of this as like a coloring book, 그러니까 색칠 공부한다고 생각하시면 되죠. So if you think of this as a coloring book, like your output, your end product should be the same size as your input product, as your input. But sometimes it is not always the case, but intuitively you want that, you want the size to be preserved between the input and the output. 준민님, 혹시 세그멘테이션에 사용하기 위한 커스터마이즈 데이터는 직접 픽셀 단위 하나하나 직접 다 나눠야 하나요? 보통 트랜스퍼 러닝 사용하나요? 혹시 세그멘테이션 사용하기? 커스터마이즈 데이터라는 게 그러니까 세그멘테이션용 학습 데이터를 말하는 건가요? 세만틱 세그멘테이션용 학습 데이터를 말씀하시는 거면은 얘기하신 대로 픽셀 단위 하나하나가 다 색칠이 돼 있어야 돼요. 그래야지 학습이 되니까. 근데 보통 이제 이 작업자들이 이 세그멘테이션용 데이터셋을 만들 때는 이 바운더리를 땁니다. 경계선을. 이제 이미지가 있고 이제 이렇게 색을 이렇게 연필 같은 마우스 이렇게 바운더리를 쭉 따요. 그렇게 해서 이제 하나의 어떤 닫힌 클로즈 셋을 만들고, 그 다음에 그럼 그 안에는 이제 다 같은 클래스로, 그 안에 있는 픽셀들은 전부 다 같은 클래스로 일단 지정을 해야겠죠. 내가 이렇게 동그라미를 친 다음에 이 동그라미 안은 전부 다 사람이다. 이렇게 네모를 친 다음에 이 네모 안은 전부 다 버스다. 이런 식으로 이제 작업자들이 작업을 해주셔야 됩니다. 답이 되셨나요? 세그멘테이션용 데이터로 제작되지 않은 데이터들 실제로 직접 나누고 있나요? 무슨 뜻인가요, 그게? 그러니까 세그멘테이션용 학습 데이터가 아니면 어떻게 되냐고 물으시는 건가요? 아니, 어떤 데이터든 세그멘테이션용 학습 데이터라는 게 태초부터 있는 게 아니잖아요. 일단 이미지가 있고 그거를 누군가 작업자가 색칠을 다 놔야지 세그멘테이션용 데이터가 학습 데이터가 되는 거니까 누군가는 항상 그걸 하고 있는 거죠. 그 작업을. 경계선 칠하는 작업을 항상 하고 있는 거죠. 답이 되셨나요? 네. Right. So the question from Junmin was that what is like the nature of the training samples for segmentation? Do we have to like classify each pixel into its distinct classes? And the answer is correct. So you need to, in order to train a segmentation model, you need to have a specific segmentation training set or training samples. And typically what you do is, as a labeler, if you're like a labeler or Amazon Mechanical Turk or something, then you would, given an image, you would draw a boundary on the image, and then inside a boundary, all the pixels inside a certain boundary that you just drew should belong to a single class. So for example, like you can draw a circle and then classify and then label all the pixels inside a circle as like a person, or you can draw a rectangle and then you can classify all the pixels inside a rectangle as a bus or something like that. So somebody needs to construct that kind of training samples for segmentation, and then you can train your model. All right, so interesting questions. All right, moving on to why segment. Yeah, if there is no more question, we'll move on to why this matters, semantic segmentation. Like why is it good? What is it good for? And the answer is there is actually a lot of diverse applications, or like something that you can actually make money with. It's not just for academia. It's not just for intellectual curiosity or something. You can actually have a business, or you can actually help people with semantic segmentation, as seen here. Obviously, if you want to develop a self-driving car, then you want your car to recognize or perceive the scene or environment around itself. So this is a front camera of a self-driving car, and obviously you want your car to distinguish between people, cars, road, crossroads, and like trees and different kinds of stuff. So obviously the most important thing that you want to recognize is obviously people, pedestrians. So as a car, you never want to collide into people. So this is actually not just image segmentation. This is actually a video segmentation. So a sequence of images is streaming into the car through a video camera or through a sensor, maybe 30 images per second or 60 images per second, like 30 FPS or 60 FPS, and you want your car to perform semantic segmentation 30 times per second. If your camera is 30 FPS. If your camera is 60 FPS, then you want your car to perform semantic segmentation 60 times per second, so that so that the things are not delayed. You're always on time. You're always like in sync with the real time. So if your car can do this, then it'll try to align with other cars while trying to avoid pedestrians or people. Another example is geosensing. So if you have a semantic segmentation model, then what you can do is given a satellite image. Some people are interested in trying to classify different types of objects inside the satellite image, like whether there's like a water or plain land or trees or that kind of stuff. So here you can you can see these rectangles are all colored for water. So I think it's water, yeah. So there's something called geosensing. The next one is precision agriculture. So again, this can also be used for agriculture, but there's something specifically called precision agriculture, like how many trees. So given a satellite image, you want to count the number of trees in a certain area, or you want to count the number of I don't know, number of animals in certain certain wild savanna, or you want to count the number of buildings inside a certain bounding box of a city, something like that. Like you want to count different types of objects given a satellite image, so that you can have like a statistics. You can have a statistical analysis of what different types of stuff. So this is another application, and this is what we're going to look at today. So organ segmentation. So given an input image, you want to distinguish or classify the pixels into different organs. So here, this is right lung, this is left lung, this is your heart, this is your collarbone, or 쇄골이죠, collarbone, and that kind of stuff basically. So here, I think this is X-ray image, and I'm not sure if there is an open source data set for X-ray based semantic segmentation data set out there. But the thing that we're going to see today is actually based on CT scan. So it's based on 3D image. But before, we're going to have to look at the model for the typical models that can perform semantic segmentation. All right, so let's, yep. So this is the end of the introduction for semantic segmentation, and UNet is what we're going to study today. So UNet is probably the most popular model for performing semantic segmentation, along with mask RCNN. So mask RCNN is a little bit different, but UNet is probably the most important or the most widely used semantic segmentation model, at least in biomedical domain. I'm not sure if it's widely used in the ordinary domain, like general domain, like dogs and cats and that kind of segmentation model. But at least in biomedical domain, UNet is probably the go-to model, like the baseline model that you want to use. And actually, UNet was indeed developed for biomedical image segmentation. It wasn't developed for organ segmentation. Actually, it was developed for a cell cancer, doing segmentation on top of a cancer cell, but still it is still like all biomedical. So the principle of doing segmentation is still the same, whether you're working on a biomedical image, whether you're working on a general domain image. It really doesn't matter, because what you want to do is actually just classify each pixel into different classes, and that's it. So given that principle, UNet is generalizable to any domain you want. So that is probably why it has 26,000 citations. So I'm not sure if there's any segmentation model that has higher citations than this. If you know, you can let me know. You can share your knowledge through on the chat box. So UNet, the name of the paper is Convolutional Networks for Biomedical Image Segmentation, and it was developed by a group of people. And the first author was this guy, or this. I'm not sure if this is a woman or a man, so I'm just going this person, Ronneberger, and it was proposed, or it was presented in MICAI 2015. So MICAI is a conference dedicated for medical image processing. So it is not general domain conference. It's a biomedical image processing conference, and it was developed way back in 2015. Way back. So this is a really old model, but still it is like the go-to model. It is such a well-made model that it is still relevant to this day. And as I said, very popular semantic segmentation model, along with mask RCNN. So mask RCNN was developed for general domain pixel segmentation. And mask RCNN is actually you can use mask RCNN. So the mask RCNN consists of two parts. The first part is faster RCNN, which is based on object detection, which performs object detection. And once you've done object detection, you push those information along with the original image into a convolutional neural networks to color the pixels inside the bounding boxes. So when you're coloring, you can use UNet to color. So those are like UNet and mask RCNN are not, it's kind of an orthogonal approach. And I didn't say it here, but UNet is a fully convolutional neural network. There's not a single fully connected layer inside UNet. So which means just like inception model or ResNet, any sized image can be used as an input. It doesn't care, because as long as when you have a fully connected layer somewhere in your convnet, then it means that you're assuming that your input image would have a fixed ratio, like fixed resolution, like 128 or 120 by 128, 256 by 256. You're assuming that your model will always receive or accept a fixed resolution. But if your model is fully convolutional, like there's zero fully connected layer in your model, then it means that that your model is flexible enough to just work on any type of the resolution, any image images of any resolution. UNet is a fully convolutional neural network, and it is based on compression and decompression architecture, or you could call this as an encoder decoder architecture as well, because encoder and decoder is kind of like compression decompression. Right, and because the output must be, so this is important, because the output must be the same size as the input, as that was question from Ijewan Nim. Yeah, so so that was a very relevant question. So the output must be the same size as the input, and that is why we need a decompression stage. If you're just doing image classification, then all you need is just compression. All you need is encoding. You have an image, and then you encode it by downsampling using convnets and max pooling or average pooling, and then you compress your image spatially while increasing the depth of your feature, and then finally you concatenate them or whatever, and then classify them into different classes. So it's just that is pure compression. But since this semantic segmentation requires the output to have the same size as the input, there must be a decoding process, or there must be a decompression process. So that is why we have a compression decompression architecture. Oh, by the way, so the assumption, the prerequisite of this class was that you went through basic machine learning and some linear algebra or calculus. That besides that, my assumption was that you went through the programming for AI class that I taught last semester, and during that class, I basically covered the basics of convnet and some different implementations of convnet, such as like VGGNet, AlexNet, Inception, and ResNet. So that was the extent of the convnet, but UNet is something a bit more different than what I've taught during the class last semester. So we're going to cover UNet a bit more in detail. So yeah, as I said, it's a compression decompression architecture. So your input looks like this. So input image tile, or just flattened image. Your input image should be, I think in here, the assumption, I'm not sure if you can see it, but the assumption of this original UNet was that the input image has a resolution of 572 by 572, right? And it has a single channel. I guess the original image was black and white. So I guess that's why they assume that your input image is 572 by 572 by 1. And then you compress it. So using going through a sequence of convolution and max pooling, you compress your image spatially. You compress your image spatially, all the way down to 30 by 30, and the depth is increased to 1024. So depth 1 was increased to 1024, and then that's the end of compression, and then you want to decompress it. So this is decompression stage. So based on the compressed image, you decompress it all the way back to not exactly the same size as the input. In here, you can see the output is 388 by 388 by 2. 380. So that is not exactly the same size as the input. And I guess I'm not sure why they did so, but they could certainly have designed their model to output the same size image as the input. I'm not sure why they just chose to use a slightly smaller output image. But you can still do a segmentation as well. You can still do segmentation based on your a little bit smaller image. All you need is just some humans to evaluate whether your output image is correctly segmented or not. Right? Okay. And so I mean, so this is why it's called UNet. This kind of like shaped like a U here. So like this, half of this U is compression, and the other half of the U is decompression or expansion. And so compression is done by sequence of convolution plus max pooling, and decompression is done by a sequence of transposed convolution. So this is what we're going to look at a bit more in detail later. So transposed convolution is a way to upsample your input, your feature map. So your feature map looks like your feature map is 30 by 30 by 1024, and you want to upsample all the way back to here, and that's where transposed convolution is involved. Yeah, so upsampling. So we're going to talk about upsampling. So this is downsampling typically. So this is Linet 5. and your input is 32 by 32. I guess this is MNIST. So 32 by 32. Actually, no, MNIST is 28 by 28. I think SciFar was 32 by 32. Anyway, so your input is 32 by 32, and then once you do a convolution without padding, then you end up with 28 by 28, and then you do subset, you do max pooling, like max pooling of size 2 with stride 2. So this becomes like 28 from 14 to 14, and then you do one more convolution. This becomes 14 to 14 to 10 to 10, and then you do again max pooling, becomes 5 to 5, and then you concatenate everything together, and then you put it through a logit layer, and then you get the classification output. So this is down sampling, and so there are a lot of different ways to do upsampling actually. So how do we upsample? You can either use bilinear interpolation, you could use bicubic interpolation, or nearest neighbor interpolation. So these three are well-known interpolation methods, and today, or actually not today, but what UNet chose to use is called transposed convolution. I think like modern GANs, like style GANs, they just use interpolation methods. So the difference between these three and this one, so these interpolation methods and transposed convolution, the difference between the two groups is that these interpolation methods do not require any learning. So there is no learnable parameter involved in these processes. But here, transposed convolution, this does require a training. So it means that this involves a learnable parameter. And actually, this is kind of like the opposite of convolution. So there are filters involved in transposed convolution, and you need to learn the parameters of the filters. So how this is like a diagram of how different interpolation works. So this is your original image. So it's like it's a one-dimensional data, and so all you have is just your x-axis and x-axis, and each location you have a height. So here you know that the height is this, and here you know the height is here, and you know height is here, here, and you want to upsample it. It means that you want to have a denser data, or denser samples. So if you use, for example, like sine, what is sine CX? I'm not sure what this sine CX means. It's I guess this is some kind of like a combination of different sinusoidal signals, like a Fourier series or something like that. So if you have this, then you can, given this unit signal or unit impulse, you can slide this unit signal along the x-axis and try to interpolate what would be the actual height here, like anywhere besides the given location. Like you want to draw basically a line like this. I'm sorry, like actually I think I can annotate. Yeah, you want to draw a line like something like this. Basically you want to have a function instead of samples. And if you have a unit impulse that looks like this, then you can slide it like doing an actual convolution. So the convolution that we do with ConvNet is discretized convolution. But if you're familiar with this like signal processing, then you know what the actual convolution is. So if you do an actual convolution, then you might get a nice looking signal like this. If you have a unit impulse, like a rectangular shaped thing, and you slide this along this axis, then you would have a very signal that looks like a step function, like this. And this is what we call a nearest neighbor interpolation. Linear interpolation, like a linear interpolation, looks like this. So you have a linear, like a linear like a weight sum type. So if your unit impulse looks like a triangle, then that amounts to like weight summing your neighbor. So if you slide this unit impulse along this axis, then you would have a slightly better looking interpolated output. So this is better than this. You know, like this is a bit more to the ground truth. If you have a Gaussian impulse noise, then you can slide this, and then you could get like an even more smoother output than linear interpolation. So again, so the important thing is that none of these methods require relearning. You just need to define what your unit impulse looks like, and then you can slide them across your x-axis. And if this is, so this example was done for one-dimensional input. So you have just x position, and for each x position, you know the value, which is the height. But if you have an image, if you have an image, then let's say that you know the values of value at here, here, and here, and here, and you want to know the value at here, for example, like here. So then what you need to do is you need to interpolate, first of all, based on, first you need to interpolate for one axis, and then you need to interpolate on another axis. So it's a sequence of interpolation. So I guess that's why they call it bilinear interpolation. So you first need to interpolate between these two pixel values in order to obtain this. And then you do interpolation for this in order to obtain this value. So this is about like 2 to 1, and this is also about 2 to 1. So you just weight sum them based on this ratio, and then once you have these value derived or interpolated, then you need to interpolate on this axis in order to get the final value here. So this is, again, this is about like 2 to 1 ratio. So you interpolate them again based on this ratio, so that you do linear interpolation twice in order to estimate or approximate a pixel value that you did not know before. And by cubic interpolation, so I'm just going to erase everything here. And by cubic interpolation is you put, instead of doing a linear weight summing between two locations, you actually put those values inside like a polynomial function to get an even smoother output. So you can actually see the examples here. So you have a very small image, like right here. It's actually an image of a bee on top of a flower. So, and you want to blow the resolution up by 10 factors. So you want, if this is like, I don't know, if this is 10 by 10, then you want to blow it up to 100 by 100. And when you use nearest neighbor interpolation of this original image, then you would get an image that looks like this. So that means that if this was 10 by 10 and this is 100 by 100, it means that your 10 pixels here. So you actually, let me. So you can see there are 1 and 2 and 3. Like there are different columns, basically. Like there are columns here. Like there's like a box here, and there's like a box here. So they all consist of pixel of the same value. So that is what I mean. This is exactly like. Oops. This is because your because you're using a rectangular shaped unit impulse. That is why you end up with a box shaped it's like blowing up a pixel by 10 by the factor of 10, basically. So that's that. And if you look at bilinear interpolation, it is way smoother than nearest neighbor interpolation. And if you see bicubic interpolation, it is even more smoother. But whether you like smoother or not so smoother image, that's up to your, I mean, it's your preference, or it's up to this depends on the task, like which one you should use. All right, so moving on. And okay, so, actually, let me just clear this. All right. So transposed convolution is a learnable upsampling process. So unlike linear or nearest neighbor bilinear bicubic interpolation, which doesn't require any learning, transposed convolution does require learning process. And So in order to in order to understand transposed convolution, you need to, first of all, obviously you need to know what convolution is, because transposed convolution is like a reverse of plain convolution. So example, we're gonna run with the example where input image is 4x4, and you're using a 3x3 kernel. And if you don't have any padding outside the image, then you would end up with 2x2 output. So like once you have your kernel applied to this patch, and then another time, next time your kernel is applied to this patch, and then next time your kernel is applied to this patch, and then finally your kernel is applied to this patch. So your kernel is applied 4 times, and that is why you have 1, 2, 3, 4 output, right? So very, very simple. And then, so yeah, this is just a diagram of the procedure of using the 3x3 kernel on the 4x4 input. So it's just basically what you're doing is just weight summing weight summing based on the patch and the weights. So you given the patch has an input signal, and your kernel has its own weights, the learnable weights, and all you're doing is just weight summing each pixel inside your 3x3 patch. And that's it. So if you think about it, if you think about this process, where you have a 3x3 kernel, and what you're doing is your 9 input pixels. So 9 input pixels, because your patch is a 3x3 patch. So there are 9 values inside your patch. So 9 input pixels, or 9 input signals, contribute to 1 output pixel. So your 9 output pixels here contribute to this guy. Another 9 input pixels contribute to this 148, like such here and here as well. So that there is a many-to-one relationship between the input, between the input to the convolution and the output of the convolution. There is a many-to-one relationship. So that is convolution, and you can think, obviously transposed convolution is the reverse of that. So what you want 1 input pixel to contribute to 9 output pixels, assuming that for transposed convolution you're using 3x3 kernel. In that case, you want 1 input pixel to contribute to 9 output pixels, so that you have a one-to-many relationship. So this is your input now. So this is your input, 2x2 input image, and you want this pixel value to contribute to 9 pixel values inside this patch, and this pixel value to 9 pixel values inside this patch. And obviously there is a filter involved. So there should be a filter that is multiplied to 2 and blows that up to 3x3. And there's the same filter, same 3x3 filter multiplied by 1, and then the output value is something. It is not exactly this, because you want this part. You want actually, let me draw this. You want this part and this part to be summed up. So this value doesn't exactly amount up to this, because but this value has exclusive impact or influence on this portion. And obviously this value, why is it not drawing so well? So this value, it has a exclusive influence, at least on this part. Actually, no, that is not correct. I'm sorry. So because that is incorrect, because this part need to be summed up with this part. So okay, I'm sorry about that. So let's draw it again. So at least this guy, when multiplied to a 3x3 kernel, has a unique influence on, or exclusive influence on this part, and this part have unique influence on this part, and this part has a unique influence on this part. This input pixel has a unique influence on this output pixel. But all the other parts are summed up with the other influences from the other pixels. So, but still, you can think of this as a one-to-many relationship. All right, moving on. Where's my? Okay, right. So why is it called transposed convolution? I mean, why is it not called like reverse convolution? So that this diagram kind of draws a nice picture for a better understanding. So if you think about a 3x3 kernel applied to a 4x4 input, which was what we did so far. So 3x3 kernel applied to 4x4 input can be represented as below. So you have your kernel, but you can spread them out like this, like here, like as a 4x16 matrix. So if you have this, then you can simply just do a matrix multiplication between this guy and your input image, flattened input image. So instead of actually sliding this guy along your 2D image, you can just instead convert this into 4x16 matrix and then simply do a matrix multiplication between this guy and the flattened image, and you get the output. And this output is 4x1, and you need to rearrange them to 2x2 in order to actually get the 2x2 output. So, I mean, this is obviously true because your input, sorry, let's see. Your kernel weights are distributed like here, here, here. So it's so the 3x3 kernel weights are like, distributed in this way. And then the same 3x3 kernel is again copy pasted here, 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 and again here, 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 so on and so forth. So they are carefully located. They're carefully assigned to this 4x16 matrix, so that the effect of I'm losing my mouse. All right, so the effect of matrix multiplication amounts or is equivalent, has the same equivalent effect of doing a convolution on your image. So if you do a transpose so if you simply do a transpose between transpose of this matrix, so this was 4x16. If you transpose into 16x4 and then do a matrix multiplication of the 16x4 in convolution weights or convolution matrix with a 2x2 or 4x1 image, then this will blow up this image into a 16x1, which you can rearrange into 4x4 image. So basically what you did is you took the 2x2 image, did a matrix multiplication with this guy, and then you got a 4x4 image. So that is, again, this is like goal achieved, right? So you can now understand why it's called transposed convolution. So you transpose this convolution into this matrix, so that the effect is, instead of downsampling, you're now upsampling. Right. All right. So let's get back to the architecture. So the half of you was confident with a convolution with max pooling, and the other half was upsampling. So now that we know how to do upsampling with transposed convolution, we'll take a look at the entire architecture. So this is as I said, this is the original architecture, but since the input and the output sizes are a bit different, and this has it's a bit, I mean, so it's not as easy as it looks to understand this architecture. So the same blog here, the author of this blog actually has a better detailed explanation or a schematic of UNet. So this is so half of the U is here, and the other half of the U is here. And it has a bit more detailed explanation. So let's take a look at them. So it starts with a plain 128 by 128 by 3 image. So we have a colored image instead. So it's an RGB image instead of a black and white, like black and white, which was used in the original UNet paper. So 128 by 128 by 3, and it goes through. Yeah, somehow when I'm trying to draw stuff, I just lose my cursor. So it goes through 2 consecutive convolutional layer, where the convolutional filter there are 16 depth and 3x3 size spatially sized filters. So, and the padding is the same. So that means that output of this guy, if you use these filters to do convolution on this input, then you would get this. So output is 16, output 16 because of this guy. Output is 16, and the padding is the same. So 128 is preserved to 128. And then you do max pull, and the max pull, the filter size is 2 by 2, and the stride is 2. So you exactly get the half of this image. So this 128 by 128 by 16 becomes 64 by 64 by 16. And then you do again, you use 32 depth 3 by 3 filter. So and then you do this two times, two convolution. So that 64 by 64 by 16 becomes 32. So 64 by 64 by 32, and then you do max pooling again, so that you get half of the So the process is such that each time you go through a convnet followed by each time you do a convolution followed by max pooling, what the effect of that is that your image becomes half the spatial resolution becomes the half, but your depth becomes the double. So 128 by 128 by 16. Where's my cursor? So 120 by 120 by 16 became 64 by 64 by 16, and then finally 64 by 64 by 32. So I'm sorry, I'm losing my mouse. All right, so yeah. So you have 120 by 120 by 16 becomes 64 64 by 32. This becomes 32 by 32 by 64. This becomes 16 by 16 by 128. So this is the end, and finally you end up with 8 by 8 by 256. So your original image 120 by 120 by 16 becomes actually 120 by 120 by 3 becomes 8 by 8 by 256, and this is the end of the downsample. This is the end of the half of the U, and now it's time to do upsampling. So you went to the bottom of the unit. Now upsampling. So you start with 8 by 8 by 256, and the upsample is such that the transposed convolution makes your image double the spatial size and half the depth. So your 8 by 8 by 256 becomes 16 by 16 by 128. Right. So this is the effect of the transposed convolution. And since you want, and you can, and let me see, and you can see these gray arrows that indicate a residual connection or skip connection. So what they want is that they want to convey the different sized encoded image or hidden representation or hidden feature map. You want them to have a helpful effect when going up. So when going down, you preserve them and then do a skip connection to the output at the output stage or at the upsamples during the upsample process. So this input is conveyed to this part, and this input is conveyed to this part, and this input is conveyed to this part. And the way they do skip connection is actually not addition. They actually do a concatenation. So you have 16 by 16 by 128 right here. Like this is the output of the first transposed convolution. You concatenate this with this here. So 16 by 16 by 128, 16 by 16 by 128. You don't do sum as the original resonant. You actually do concatenation. So that is why you end up with 16 by 16 by 256. So once you have this, you put it through another transposed convolution. Actually, no. Once you have this, you do a convolutional neural network. So once you have a upsampled or blown up image, you do a convolutional neural network. You do convolution. So you apply 128 depth and 3 by 3 filters. So this is changed. 16 by 16 by 256 is changed to 16 by 16 by 128. So there's 128 depth here. So this becomes 128. And then you again do a transposed convolution. So half double the space and double the spatial resolution and half the depth. So 16, 16, 120 becomes 32, 32, 64. And then you concatenate this with this guy so that it becomes 30 by 32 by 128. Again, 2 consecutive convolution. So 32 by 32 by 64. So you do this again and again until you are left with 128 by 128 by 16. So this is where you started. So this is where you started. And then you finally do a 1-by-1 convolutional filter in order to classify your final input, in order to classify each pixel into different classes. Here, the final output is 128 by 128 by 1, and the reason is because I think that the author of this blog was just doing a simple binary classification. So all you need is just one output, just a sigmoid output. So your 128 by 128 by 16 is converted to 128 by 128 by 1, and each pixel in this final output indicates whether a single pixel is like true or false, basically binary. So hopefully this is pretty much clear, you know. This is nothing but just a sequence of convolution, max pooling, and transposed convolution, and that's it. And you know already what transposed convolution is. So hopefully, once you see this diagram, you can write this out in PyTorch or TensorFlow for that matter. All right, and so the important thing is that the final operation with one by one convolution. So here, the original in the original figure, it was this part, and the more detailed figure, it was this part, right? And use this. So the final one by one convolution is to use this process or use this phase to produce the desired number of classes. So this is kind of like, you know, kind of like when you have a input feature. I mean, when you have an image classification, you have a final like global average pooling or whatever, and then you have a feature, and you want to map that feature into logits. Like if you want to do, if you're doing ImageNet challenge, then you want your global average pooled feature map to be divided into 1,000 classes. Or if you're doing MNIST, you want your final feature to be converted to 10-dimensional logits, something like that. So here you have 128 by 128 by 16, which is the final feature map, and you want individual pixel, which is 1 by 1 by 16. You want that to be converted to 1 by 1 by output class, number of output classes. So if your number of output classes is 10, then you should convert your 128 by 120 by 16 to 120 by 120 by 10, so that each depth or each channel corresponds to a single class, like a class in your entire 10 classes. Hopefully this is clear. Right, so training unit is pretty simple. It can be done with just cross entropy loss, because you're just basically doing for each pixel a multi-class classification. So all you need is just just add a mean cross entropy loss, and that's it. But there are some a bit more advanced loss functions, such as IOU, intersectional reunion, or Jacquard index. This evaluates how much there is an overlap between the true segmentation and your predicted segmentation. And obviously, you want this and this part to be the same. You want your IOU to be 1 actually. And there's something a bit more popular than IOU, which is called dice score. So this is dice score, and this is basically just an F1 score. So this is your true segmentation. This is your predicted segmentation, and you just add the number of pixels in each segmentation, and then you calculate the number of overlap between the predicted and the true, and then you do you multiply by 2, so you get the harmonic mean. So this is dice score, and people use I think the original unit used just cross entropy loss, but there's like some advanced units such as like NN unit, or other people use like different losses. And I think a bit like people sometimes use a sum of entropy loss with dice score. It's like entropy loss plus dice score with weights controlled, or maybe just with 1:1 ratio. Right. So the unit was originally the author of the unit trained the unit for ISBI cell tracking challenge. So ISBI is International Symposium of Biomedical Imaging. So they have a challenge every year. I'm not sure if it's every year, but when this paper was written, it was 2015. So they had the ISBI cell tracking challenge 2015, where unit won the first place. And this is the example. So, given an image, given like a cell image, you want your unit model to have to like segment different cells. So the yellow line is the ground truth boundary, and the blue pixel is the predicted pixel by unit. And it is doing a pretty good job actually. This is another example here. There are a lot of cells, and you want the different cells to be segmented in different locations. And here, the yellow line is again the ground truth segmentation or ground truth boundary, and the blue and the colors are what UNet predicted. And it is not super exact, but it is still very much, you know, visible. Like if you are a person, if you're like a biologist or whatever, and when you see a colored image like this, then it is way better than seeing a non-colored image like this. Like compared to this and this, it is way more helpful to see this guy, right? Hopefully. Right. Okay, so this is the end of UNet, and you can obviously use UNet for a lot of different stuff, but I'm just gonna introduce one more application, which is multi-organ segmentation. So specifically multi-organ segmentation using CT scan images. So it's CT scan images, meaning that you have a 3D image. So you have a sequence of images, sequence of 2D images along the z-axis. Z-axis being, you know, being like your height. So one representative data set example is multi-atlas labeling beyond the cranial vault, which is just MAL. So MAL, MAL data set. And in the MAL data set, there are different categories, but we're going to look at the abdomen data set. Abdomen is 배죠, 배. 복부입니다, abdomen. So what we want to do is to segment 13 different organs inside your abdomen, inside your stomach. So they're like spleen, right kidney, left kidney, gallbladder, esophagus, liver, you know, like artery. What else are there? There's like pancreas, something like that. So it doesn't. So heart and lung not included. So heart and lung is chest. Abdomen is abdomen 복부니까 폐 아래부터 시작한다고 보시면 됩니다. 이제 간 정도나 뭐 간 약간 위에서 시작한다고 보시면 되고. 그래서 13개의 그게 organ을 이제 segmentation 하는 게 이 데이터셋의 목적입니다. 그래서 이거 보시면 뭐 이렇게 생겼어요. It looks like this. So spleen. So this is what actual spleen looks like, and the data set. If you only look at this, you know, along the z-axis, then what you would see is this. So you have this is like a single slice along your z-axis. So there would be like, you know, at least tens, maybe 100 sequence of slices. So your spleen looks like this, your right kidney, your left kidney look like this, and you can see a very small, very, very tiny segmented pixels here. So you want your UNet model to have, since there are 13 organs, you want your UNet model to have 13 output classes, right? So input image, the spatial resolution should be the same, but the depth should be 13 instead of 1. I guess this is 1 because it's a black and white image. And so some are a bit doable. Like spleen is a pretty large organ, so it is doable. But kidneys first of all, they're not linearly shaped. They're like U-shaped, so it's a bit challenging. If you look at, some are really simple. So this is inferior vena cava. I'm not sure what this is. This could be like a blood vessel. So anyway, this has a very long, it's like an elongated rope. And so the slice looks like it's just like an ellipsoid or a circle. So it is way more doable than complex, like very weird looking, like, you know, what is this? This is portal vein. Yeah. And this is pancreas. So pancreas looks like a 기역자처럼 생겼죠. So it's very compared to spleen and like adrenal gland, which is like spherically shaped. This is way more complex. So if you think about slicing along the z-axis, their shape would change very dramatically from slice to slice. Like if you think about the slice here and here, in the lower slice there will be just one patch of green pixels, but in the upper slice there will be 2 regions with green pixels because they're like, you know, split into different regions. So at some point there should be like 1 green pixel here and 1 green pixel here. Same goes with this. Like this would, if you think about the 2 different slices here, it would just have just one circular thing here. But if you're looking at here, then it would look like kind of look like this. So it dramatically changes from slice to slice, and it really is confusing for the UNet model to do a like correct and clean segmentation for this type of organs. All right. Okay, so yeah, this is the, I think this is, yeah, this is the end of the class. So so hopefully if people are interested in organ segmentation or cell segmentation, cell tracking, there are a lot of different biomedical image based segmentation challenges, segmentation data sets, especially for brain tumors or like pathology slides or biopsy slides. There are a lot of different samples, and if you need to perform segmentation, then you should probably start with UNet and then work your way towards like 3D UNet or 3D VNet or whatever. Like there are a lot of different variants of UNet out there. All right, any question? All right, I guess if there are no more questions, then I'll see you guys next Tuesday. So have a good weekend and bye-bye.